Hello everyone and welcome to Instant Biology by Dr. Neelab. So today what we are going to start with is a very important and a very requested topic. So the topic that we are going to deal today is DNA extraction through various methods. So we are going to talk about the DNA extraction today, and there are different types of methods which can be used for the purpose of DNA extraction. We would be talking about that also. And those methods would be basically to extract the genomic DNA, because there can be plasmid DNA also, but what we are aiming at is taking out the genomic DNA. Okay, so I would be generally talking about the various methods of DNA extraction. So there can be different methods. I will be telling you about each one, the salient features about each one of them. So starting with, what are the different types of methods for DNA extraction? So the first method over here is organic extraction. The second one is silica based method. Third one is magnetic bead based method. Fourth one is anion exchange method, and the final fifth one is C-tab based method. So these are the different types of methods that are used for isolating the DNA. Okay, and what or when are they used, or what are the salient features on the basis of which these methods differ from each other? So the DNA that you get from each one of these methods differs in terms of yield as well as purity. Okay, so each method has its own positives and negatives, and because of that the yield and purity of the DNA that is obtained from these methods can vary. So let us start with the first method. This is the most common method. This is mostly used in the labs nowadays. This is the organic extraction method. So this is the organic extraction method, and I will be telling you about this method with the help of a protocol. Okay, because many students ask what is the proper protocol for DNA extraction. So I will be telling you with the help of this particular protocol. I would also be telling you about the utilization or the uses of different chemicals that are involved over here. So first thing is, harvest cells from the overnight grown culture by centrifugation at 8000 rpm at 10 degrees centigrade for 15 minutes and discard the supernatant. So over here I am showing you the organic extraction method with the help of bacterial cells. So suppose what I am trying to say is, suppose this is the petri plate on which your bacteria are growing. These are the bacteria that are growing on the petri plate. What you need to do is, you need to isolate, or you need to take out all of these bacteria, and these bacteria, mind you, have been grown overnight. These bacteria have been grown overnight, and you harvest them. That means you take out all the colonies from this and you put it inside a tube or something like this. Okay, and do a centrifugation. Okay, what you do is centrifuge it. So what you can do is, you can use a buffer or something in which you put your these cells. Okay, and after that you centrifuge it. What should be the centrifugation criteria? It should be 8000 rpm at 10 degrees centigrade for 15 minutes. So what will happen? You know what will happen is that the precipitate would form after you centrifuge. What will happen, if this is the tube, there would be centrifugation. After the centrifugation, there would be the precipitate that would be formed. This precipitate would be representing your cells. Okay, so these would be the cells, and this method, or basically this step, is separating the unwanted materials from the medium. Separating the unwanted materials from the medium, that means you just want the cells to obtain the cells only, because inside the medium where these bacterial cells would be growing, if you try and centrifuge it, then what will happen, the cells would be separated from all the media and everything else, all the contaminants, so you would be getting the cells over here. So this was the purpose of the first point, or this was the explanation of the first point. Now let us move on to the second point. Wash the pellet twice with normal saline and centrifuge at 8000 rpm at 10 degrees centigrade for 15 minutes. Again, what you have to do is you have to wash the pellet. So the pellet that you had obtained over here, so this was the pellet, and pellet comprised of the cells. So you had your pellet, and pellet basically comprises of the cells, and there might be some other contaminants also that still have not been able to get removed because of the centrifugation that was done. Still some unwanted materials or some undesired materials or some contaminants are present over here. So suppose that there is some contamination over here still. What you do is you wash it with normal saline. Normal saline means NaCl. You wash it with the normal saline. Wash with normal saline, and then again centrifuge it. Again after that you centrifuge it. Now the purpose is to just enhance the concentration of our cells, to reduce the contamination and reduce contamination and enhance the concentration, enhance the purity of the cells. That is why we washed it. That is why there was washing of the pellet, and also again you perform the centrifugation at 8000 rpm and 10 degrees centigrade. This was the second method, or this was the second step, sorry. Now the third step is add 0.5 ml of 10 millimolar Tris Hcl and 2.5 mg per ml of lysozyme and incubate at 37 degrees centigrade for 1 to 2 hours. Now you have your cell, that means you have the cells. Now what you are doing is you are adding Tris Hcl. What is the function of Tris Hcl? Let us understand this. Tris Hcl is actually acting as a buffer to maintain the pH. It is just required for maintaining the pH. Otherwise the cellular content that we are having, it might get destroyed or whatever we want. We want the DNA, right? So before you obtain the cells, you have to maintain an environment so that the cells remain intact. Just for now, the pH remains intact just for now. Okay, so because you are isolating the cells just now, and you would be interacting the cells from now with different types of materials, so for now you want to maintain the pH, and this work or this maintenance of the pH is performed by Tris Hcl. Okay, next what you are doing is you are also adding lysozymes. So what are lysozymes? Lysozymes are enzymes that are hydrolytic in nature. These are enzymes hydrolytic in nature, and this would basically break down the cell wall, break the cell wall of the bacteria, because we are working on the bacteria, right? So the cell wall of the bacteria would be broken down, and you have to incubate this with the Tris Hcl and the lysozyme. Okay, so this completes the third step. Okay, just one more thing for those of you who do not know, that this suppose is the cell wall, or suppose this is the cell membrane. Just outside the cell membrane there is the cell wall, and in some cases a membrane outside the cell wall is also present. This is called as glycocalyx. It is also called as glycocalyx. Okay, so what we are intending, we want to remove the cell wall glycocalyx with the help of lysozyme. Okay, so now coming to the fourth point. The fourth point states that you have to add the lysis buffer. Now what is the function of the lysis buffer? Lysis buffer, as the name suggests, causes lysis. It causes lysis. Lysis means breakdown of the cell or disruption of the cell. Okay, and what are the main components of this lysis buffer? Let us understand this. The main components of the lysis buffer involve Tris. Over here it is written Tris, plus it has EDTA plus it has SDS. Okay, so these are the components of the lysis buffer. So first of all, let us talk about the Tris. What does Tris do? Tris basically is used for solubilizing the nucleic acids. Okay, so what we are working on is, we are working on the DNA only, right? So you would want to solubilize the DNA first of all. Okay, because if it remains in the precipitate form, it might get the concentration might get less, something can happen. So what you want to do is, you want to solubilize it. So Tris basically helps in solubilization. Next, let us talk about EDTA. What is the function of EDTA? Let us talk about that. Just a minute. Yeah, EDTA. So you might have heard that EDTA is the chelating agent. So what does a chelating agent do? It chelates the metal ions. Why am I saying chelates the metal ions? You might be knowing that the DNAs or the nuclease, let us talk about the DNAs. These DNAs, what will they do? They will cleave off the DNA. These DNAs require Mg2 plus as the cofactor. Without this, they would not be able to function. So we would not want DNAs in our solution, right? Because if the DNAs would be present, it will definitely break down the genetic material or the DNA that we want to isolate. That is why we add EDTA. EDTA, what will it do? It will chelate the Mg2 plus, that is, it will not allow the free Mg2 plus to be accessible by the DNAs. Okay, once the DNAs is not able to access the Mg2 plus, it would not be able to function. Once it does not able to function, what will happen? The DNA will not get destroyed. Okay, so the DNA will be safe, it will not get destroyed. Next is SDS. You might be knowing that this particular SDS, or the sodium dodecyl sulfate, is a detergent. And you might be knowing that these detergents, why are they so useful? They are useful because they solubilize the phospholipid membrane. So what will happen? The phospholipid membrane, this is the cell. Suppose we have already broken down the cell wall with the help of the lysozyme. Suppose this is the cell membrane now. Okay, this is the cell membrane. Now you want to remove the cell membrane. So what will happen? SDS will act on the cell membrane and remove it, because SDS is a detergent. The DNAs that was present, the Mg2 plus ions that were present, they would be taken up by the EDTA. So that would also not be useful. Now everything that is present in the solution would be safe. That is the DNA that is present in the solution. This is the, let us say that this is the DNA present inside the solution would be safe because of the Tris, EDTA and SDS that is being used over here. After Tris, EDTA and SDS, these were the components of the lysis buffer. We have to mix proteinase K. Now as the name suggests, what is this proteinase? The proteinase simply means that it will cleave off the protein or distort the protein or destroy the protein. Okay, so cleave the proteins, because you would be knowing that inside any cell there can be protein also, there can be nucleic acids also, right? So we do not want any proteins or any contamination of the proteins in our DNA. So that is why we are ensuring that all the proteins, they get cleaved off or they get destroyed after interacting with proteinase K, and that is the reason why this proteinase K have been added. So with this we complete the fourth step. Let us move on to the fifth step. Okay, so after this digestion with the proteinase K, what you do is you add phenol chloroform in the ratio of 1:1. Phenol chloroform method. Now you might have also studied about phenol chloroform method. Now this phenol and chloroform, now these two are immiscible liquids. Why am I saying immiscible liquids? Because the nature of the phenol and nature of the chloroform is entirely different. Chloroform is a aqueous solvent, whereas phenol is a non-aqueous solvent. Non-aqueous. Okay, that is the reason why when you mix phenol and chloroform, it will never make a clear solution. There would always be a interface that is present. So suppose this is the interface that is present. Interface, this is the interface, and let me just for your ease, let me just put some color over here. Right. So suppose this is the region of chloroform and this is the region of phenol. Okay, so phenol and chloroform, this is the upper region, is the region of phenol, chloroform, and this is the region of phenol. Now what we are saying is, once you have chelated the Mg2+, you have dissolved the or destroyed the proteins that were present, what we are doing is we are making a phenol chloroform method, and then we put the solution that we had obtained from the above step that had our DNA, we are transferring the DNA into this solution. Okay, and then it is mixed gently for 2 to 3 minutes. After that, centrifugation was done at 10,000 rpm at 4 degrees centigrade. Okay, now what I want to make you understand over here is that DNA is polar. Why is the DNA polar? Because it has phosphate groups. Phosphate groups. These phosphate groups are negatively charged, and because of that there is polarity in DNA. Okay, so you would be knowing that like dissolves like dissolves like. That means the aqueous solution or the chloroform, because it is an aqueous solvent, it will dissolve, just a minute. It will dissolve DNA. Okay, so DNA would ultimately come over here in this particular layer. Okay, so DNA would be present over here in the upper layer or the aqueous solution of chloroform. Okay, so this is what is written over here: transfer the upper layer in a fresh sterile tube. So what we have to do is, this was the upper layer, let us say this was the interface, and this was the upper layer, right? Upper layer. So this upper layer was transferred into a separate tube, a fresh tube. Why we are transferring it into a fresh tube? Because this layer is only containing DNA. So when you take this or pump this out of out of this tube through the help of a pipette, now this is a pipette, you pipette out the top or upper layer, your DNA is also coming out with this. Okay, then the upper layer is transferred in fresh sterile tube and extract with chloroform isoamyl alcohol. Next step is you have to extract the DNA out of this layer with the help of chloroform isoamyl alcohol. Okay, so what will happen? This will cause the extraction from this particular solution, the chloroform solution, because you have your DNA present in the chloroform. From the chloroform you need to transfer it into chloroform isoamyl alcohol. The ratio should be 24:7, 24:1, what is the use of this? Because this is able to facilitate, this is able to help the isolation of the DNA. So what you are doing actually, you are transferring chloroform, you are making the you are introducing alcohol over here. Okay, so this is a solution of chloroform and isoamyl alcohol. This is an alcohol, because you are preparing for the next step, because in the next step there is 95% ethanol that we are going to treat our DNA with. So the supernatant that was obtained from this particular step, that is chloroform isoamyl alcohol, the centrifugation step, you have to repeat this step also. After this step, you have to take the supernatant and then you have to add 5 molar of NaCl, and then you have to add the 95% of ethanol over here. After this, you have to add NaCl, and then you have to add 95% ethanol over here. What will this cause? This will cause again the precipitation. This will cause the precipitation. The DNA that you had would precipitate down. Okay. Next, centrifuge again at 10,000 rpm for 4 degrees centigrade for 15 minutes and decant the supernatant. After this particular step, what we are doing is, again you are centrifuging at 4 degrees centigrade for 15 minutes, and then you remove the supernatants. After removing the supernatants, you have your pellet. Now this pellet is allowed to dry. Okay, this pellet is allowed to dry, and to because it was having the alcohol also, so that all the alcohol that was present can be removed, it is allowed to dry. Okay, so now you are having your pellet only. Now what is the next step? Now what you can do is, you can add your TE buffer. Okay, because TE buffer is only used to it is used to dissolve the pellet. So what is TE means? Tris and EDTA. So I have already told you what is the purpose of EDTA. You do not want any DNAs to cleave off your DNA, and Tris basically is used to solubilize the nucleic acid. So TE buffer is used for suspending or solubilizing the DNA material. Okay, so this will allow the stopping, or this will prevent the activity of any kind of nuclease also, plus it will allow the proper solubilization of the DNA that you had obtained. Next, what you can do is, you can check the quality or the amount of the DNA that you have obtained through the gel electrophoresis. You can do that, plus you can also use nanogram. So these were the steps through which you can isolate the material or the DNA with the help of organic solvent. I hope you would have understood this particular topic pretty well. Now coming to the next method of DNA extraction. So the next method is silica based method. This is also called as solid phase DNA extraction, also called as solid phase. Now why are we calling it solid phase? Because there is nothing liquid over here. Previously in the organic solvent, in the organic method, we had multiple different types of solvents that were acting on the DNA, but over here there is nothing but a solid phase. What is the solid phase over here? There is a silica tube you can use that is having silicates present on it, and that is solid basically, right? It is mostly in a solid form. That is why you are calling it as solid phase DNA extraction. Now how come this method is working, or what is the basic principle behind this method? So there are silicates, you know, because we are having silica, so there would be silicates that are present. And one thing about silicates is that they have very high binding affinity for the DNA, but this affinity works under alkaline conditions and high salt concentration only. That means whenever you have, just a moment, whenever you have alkaline conditions, means that pH is high, plus there are high salt concentrations also, then what will happen? The affinity of the silicates for the DNA increases. Now you can make use of this property. How can you make the use? Because the silicates at this particular concentration, they show positive charge. Okay, so once the pH is increased, salt concentration is increased, the positive charges basically emerge out of silicates. Okay, so this is something like this. So suppose these are the silicates. So these silicates would be having positive charges. Now since they are having positive charges, you would be knowing that the DNA has a negative charge because of the phosphate. This is having PO4 2 minus because of the phosphates. So there can be an interaction between the negative charge and the positive charge in case of the silica based method. I hope you would have understood this. How could you elute? Now how can you go for elution? What does elution means? You want to remove the DNA from these silicates. If you want your DNA, if you want to work on the DNA, you will have to isolate the DNA from the silicates also. You have absorbed the DNA onto the silicates, but you want to get this DNA out of the silicates also. How would you do this? What is the method through which you can do this? So you can elute the DNA, elution of the DNA by any hypo-osmotic solution. Hypo-osmotic solution. So there are two types, hyper and hypo. Hypo means that is having less concentration of the solutes. So once you are using hypo-osmotic solution, what are the examples? You can use nuclease free water. You can use buffers such as alkaline Tris Edta. So these are the two things, nuclease free water or other buffers such as alkaline Tris Edta. Through this method, what you can use is, or what you can do is, you can elute out your genetic material or the DNA from the silicates. So this was the silica based method. Let us move on to the next method, and that is the magnetic bead based method. As the name suggests, over here you would utilize magnetic beads. Now what are magnetic beads? These are basically paramagnetic materials. Paramagnetic means they are having less attraction towards the magnets, and they also have the uniform particle size. Uniform particle size. These are magnetic beads, paramagnetic in nature, and they have uniform particle size. Now these magnetic beads are coated with functional groups. Let us understand this. Suppose this is the magnetic bead. Magnetic bead. Now this magnetic bead would be coated with some functional groups like this. Okay, these functional groups, and these functional groups would be interacting with the DNA like this. These would be interacting with the DNA. Okay, but naturally it will be having some kind of charge interaction. That means the DNA is negatively charged, and the magnetic beads, the functional groups that are present in the magnetic beads would be positively charged, and because of the positive and negative charge interaction, these functional groups would be able to bind with the DNA. Next, because these magnetic beads are paramagnetic, they can be pulled out through the action of magnetic field. Now what you do, if you are using a magnetic field, what will happen? The magnetic bead will come and get attracted, or will get in contact with the magnetic field, because magnetic field, what will it do? It will pull the magnetic beads towards itself. Now once the magnetic bead is pulled, because the magnetic bead had the functional groups, and functional groups were joining or combining or interacting with the DNA, so DNA would also come along with the magnetic field. Okay, so over here, as you saw, there was no requirement of the centrifuge or vacuum. In the previous examples I showed you that centrifuge was required a lot. Now you should understand that centrifuge creates a lot of stress or shear on the material that you are able to, or you are targeting for absorption, or you are targeting for isolation. You would not want your material to get destroyed. That is why you should aim at a method which utilizes minimum amount of stress. So over here you can see that no vacuum or centrifuge was utilized. You just used some amount of magnetic field to pull the magnetic beads. I hope you understood this. The next method is anion exchange method. Okay, so anion exchange method, you might have heard about the ion exchange chromatography. In that also we have already studied anionic exchange chromatography. I have already made a video on that. If you do not know, if you have not read about that, you can just check out my previous videos. So anion exchange method, what does it mean? It simply means that you want to exchange an anion. Okay, you want to exchange an anion. What or how can you exchange an anion, or what is basically anion? Anion is something that is negatively charged. Okay, so anion is something that is negatively charged. So suppose this is our resin. Okay, this is what? This is our resin. This is our resin, and this resin would be positively charged. Remember, this resin that we are talking about should be positively charged, and to this resin, positively charged resin, there is a functional group that is temporary functional group that is bound. Suppose I am writing it as Y. This Y is a temporary functional group that is attached to this. Now what will happen when the DNA, now definitely this Y would be negatively charged because it is getting attracted to the positive end, to the positive resin. When the DNA comes along, DNA is having a lot of negatively charged phosphates. Because of that, this DNA would come and replace the Y and would bind to the resin. Okay, so this is the principle that is already being used in the anion exchange chromatography. The same principle is being applied over here also. Okay, so now what will happen is this Y will get removed from here, and this DNA will come and bind to the resin. So now presently what would be the scenario? This would be the positive charged resin, and to this your DNA would be bound like this. In this way, what you can do is you can isolate the DNA from the solution. Okay, so this was the story about this. Let us just read through the text. So over here the resins are basically positively charged. One such example is diethyl aminoethyl, DEAE cellulose. Okay, so what does it do? It attracts the negatively charged DNA. Negatively charged DNA is attracted to this because the resin itself is positively charged. Next, when at what conditions the binding takes place, that means at what conditions the DNA would be able to bind to the resins. Always remember that the salt concentrations should be low if you want your DNA to bind to the resin. Definitely, if the salt concentration would be high, then the salt ions would only bind with the resin. It will not allow the DNA to bind with the resin. That is why you want to lower down the salt concentration, only then there would be the possible binding. Next, how would you do elution? Just opposite to the binding is elution. You just have to use the high salt concentration. Okay, once you use the high salt concentration, what will happen? Once you use the high salt concentration, what will happen? This DNA would be removed, and the salt, suppose you are using NaCl, the Cl would be separated from the Na, and Cl might come and bind over here. Okay, therefore the DNA would be removed. Next, purity of the DNA is high as compared to silica. Important point. Okay, so in case of the silicates, the purity is not that much, but because of this anionic exchange or anionic extraction, the purity is high. One important disadvantage is that in case of elution, what you were using, for the elution you were using very high salt concentration. For elution you were using very high salt concentration, and that salt came into the the DNA also. That is why you want to desalt after the elution, because this DNA would be utilized for downstream processing also, right? Downstream processing also. So after elution, because this DNA would be in interaction with the salt also, you would want to remove the salts. This removal of the salt would be called as desalting. Okay, why? Because this DNA would be required for downstream processing, so you need to remove this salt. Okay, I believe you would have understood this. Now let us move on to the last method, that is called as the C-tab method. Now this is also a very common method, but it is mostly used for the plant samples. Okay, the full form of C-tab is cetyl trimethyl ammonium bromide. It's also called as C-tab method. In the same concept is here, that when you are using the low ionic strength solutions, that means the ionic strength is low, then what will happen? The precipitation will take place. Precipitation of what? Of nucleic acids and acid polysaccharides. There would be a precipitation of nucleic acids and acid polysaccharides at low ionic concentrations or low ionic strength. Okay, when if you want to this because of the low ionic strength solution, the precipitation was made, and if you want to dissolve the precipitate or solubilize this precipitate, what you need to do is you need to use a high salt concentration. When you use a high salt concentration, the precipitate that was formed of nucleic acid and acidic polysaccharide, it will dissolve, and acidic polysaccharides will still remain in the precipitate, but nucleic acid would now go into the supernatant. Very important point. Okay, when you have low ionic strength, there is a precipitate that is formed, and inside this precipitate there is nucleic acids also and acidic polysaccharides also are present. But when you increase the salt concentration, that is the high salt concentrations are present, what will happen? This precipitate would be remaining, and it will now only comprise of the acidic polysaccharides, and nucleic acids would be removed from here. Nucleic acids would be removed from this. Okay, so this was the C-tab method, and nucleic acid you would obtain. Now there are also multiple solvents that are present, that are utilized in the C-tab method. You use phenol, chloroform, isoamyl alcohol, mercaptoethanol, so on and so forth. So these are the solvents that are required in this C-tab method. We have talked about C-tab, we have talked about anion exchange, we have also talked about magnetic bead, we have also talked about silica based method, and organic extraction method also we have talked about. So these were all the DNA extraction protocols or methods. I believe you would have understood this pretty well. If you have understood this, please like the video, please subscribe to my channel, press the bell icon, and also share it with your friends so that all of you can study together. Thank you and have a good day.